Hello, welcome. I'm Garima Trimbati from RKGIT Ghaziabad and will be discussing Unit 5 topic extraction of crude drug. Here we shall be discussing maceration, percolation, modified maceration and certain specialized extraction procedures. So, as the name suggests, extraction means dissolving the solute material using a solvent system and separating it completely. So, as written, the extraction is defined as the process of isolation of soluble material from an insoluble residue, which may be liquid, solid, by treatment with a solvent on the basis of physical nature of the crude drugs to be extracted. So, depending on this, the process may be divided into number 1, liquid extraction procedure, or number 2, the solid liquid extraction procedure. So, this particular extraction procedure is controlled by the basic principle of the mass transfer. That means that the plant material having certain phytoconstituents, either primary metabolite or secondary metabolite, is gets imbibed, and the materials are available as free solutions, free cell solution, and this when passes into the solvent system and forms an equilibrium. So, the extraction method or process ends when the equilibrium is established between this particular cell free system and the solvent system. Extraction has certain purpose which we can enlist as number 1, quality control, number 2, extraction of phytoconstituent, or isolation of phytoconstituent, then analysis of these phytoconstituent. Fourth one, tissue culture. Fifth one, formulation development. Sixth one, particularly obtaining an API. So, these are the various purpose of extraction procedure. Moving to the next slide, choosing a solvent or the choice of solvent, that has to be taken care in the extraction system. So, the characteristic depends mainly on the metabolite. So, metabolite is an substance which is generated biosynthetically in a plant body. This could be either primary metabolite or a secondary metabolite. So, examples of primary metabolites are the one which is obtained from basic cycles like TCA cycle, uric acid cycle, with the carbohydrates, the proteins, amino acids, etc. While the secondary metabolites take this primary metabolite as the precursor, and then from the newer metabolites, and these are pharmaceutical importance as they are highly biologically active, showing various medicinal properties. Some examples could be enlisted as alkaloids, glycosides, anthraquinones with the anthraquinone glycosides, isothiocyanates, thiocyanates, terpenoids, steroids, etc. Okay. So, these are the various phytoconstituents or the various secondary metabolites that are isolated from the plant materials. So, these chemical molecules polarity is highly important, with this steroid is a lipophilic or lipid loving substance, and this require dissolution or solvation in a non-polar solvent like petroleum ether. Similar is the case with the phytoconstituent flavonoid, which is highly soluble in the petroleum ether extracts, or here is the case with the certain glycosides. Depending upon if the molecule is larger, it may choose more non-polar solvent as compared to the polar ones. If the phytoconstituent could be a sugar type, so it will require more of a polar substance for the isolation. So next is the pH. pH of this particular secondary metabolite is also under consideration, as the negative concentration of the hydrogen ion interacting with the solvent or the solvents is very important for the solvation of the secondary metabolites. Then comes the thermal stability. For example, if we take vitamin C of the citrus fruits, these are at or above 40 degree get easily denatured. So any solvent choice which will affect its stability, or which will require to have a higher temperature to extract vitamin C, will obviously decrease the amount of vitamin C extracted and will cause denaturation of vitamin C. Next is the ideal characteristic of the solvents that are being chosen for the extraction procedure. We must take care that it should be able to dissolve at the maximum amount of the phytoconstituent or the secondary or the primary metabolite which is intended or desired to get to extract. The solvent must be non-inflammable. Secondly, it should be inert. That means it should not be interacting or forming a newer compound or some biologically inactive compound with the interaction of the secondary metabolite. Then comes non-toxic. The solvent, since we are consuming or we are utilizing these phytoconstituents for biological purpose, so the first important, it is very important to be the solvent being less toxic in nature. For example, methanol is known to be highly toxic. So methanol extracts many a times are not preferable for various phytochemical extraction or various other extractions, and hence certain fermentation generated products or aqueous extracts are far more preferable. Okay, so moving to the next point is easy removable. So the solvent which is utilized to extract the phytoconstituent should not leave any residual solvent while that is being dried. The solvent must be removed completely and it should not have any residual solvent left on the phytoconstituent. This is again a primary requirement in the extraction, in the choice of the extraction procedure. Next topic of discussion is choice of extraction method. See, extraction methods depending upon the sample size, quantity of the extract required and the extraction time required for the extraction of a particular phytoconstituent, as well as the cost has to be considered while choosing an extraction method. Supposingly a substance which is having a hard type of plant material or animal product, so such substance has to be powdered and powdered coarsely or moderately or finely to get the desired sample size. A coarse powder must pass through the sieve size of 10 by 40 mesh size, while the moderate ones must pass through 20 by 60 or 20 by 80 20 by 60 or 20 by 80 as required. Okay. Next is quantity. Obviously, the amount of extract required depends upon the usage, whether it is used for some biological activity research or just phytochemical analysis or isolation or preparatory purpose. Now, the term preparatory has to be explained. This term preparatory refers to a condition where during the manufacturing or during the research, when we need a high amount of the given phytoconstituents, So, mainly it is to obtain larger amount of the phytoconstituent as well as to purify the APIs when they are isolated for certain analytical purpose. Okay. Moving to the next topic are the factors affecting the extraction of the crude drug. So, enlisting the factors are quantity and nature of the crude drug. Next is size of the powder of the crude drugs. Next is moisture content of the crude drugs, nature and volume of the solvents. Next is the mixing ratio of the solvents and the sample. We shall discuss them, we should illustrate them or discuss them in the further presentation. So, another important factor here is the temperature of the extraction procedure, pH of the extracting solvent as well as the lipophilicity. So, here I must take the privilege to explain the moisture content of the drug. So, what happens here is any plant material which is being chosen, either the plant part is dried and utilized for the drug product, or it has its fresh material is used for the extraction procedure, or else some unorganized material, unorganized part is used, or sometimes these animal parts, animal products are also utilized for the obtention of the secondary metabolites. So, in dry material almost 3 to 5% of moisture content is available. Here more than 12% or 15% you can say is available, and unorganized drugs, the moisture content variability is there. Okay, now this moisture content means presence of water, and when in along with the solvent, it will cause the partitioning or the distributioning of the solute that is intended for extraction, and hence the extraction will be limited or lesser efficient. So, the consideration of moisture content is very important. Next is mixing ratio. As communicated here, there are many materials or solvents which cannot easily penetrate into the cell wall, but are capable of diluting or dissolving the solute in the cell material. So, if we change the little ratio of the solvent system, that allows the penetration as well as solvation of the solute, and hence this mixing ratio is an important consideration in the extraction procedure. Then comes temperature of the extraction procedure. Again here I will highlight one point, that is like cardiac glycosides. See, cardiac glycosides, so these cardiac glycosides are generally extracted in a cold condition. Why? Because they are highly sensitive to the temperature, and the glycon and aglycon separates, and hence it the biological activity of cardiac glycoside is affected. So, extraction we take utmost care while extracting the cardiac glycoside. Similar is pH of the extracting solvent. Again, interaction or hydrogen ion concentration of solvent and the interaction of these hydrogen molecules with the solute affects the extraction procedure. Then is another very important point for consideration is the lipophilicity of the substances. So, now there are various substances that we are considering in our extraction. One is the sample itself, either it could be a lipid loving solvent, it could be lipid loving or lipophilic. Then we have the solvent which itself is lipophilic, okay, and there are certain interfering materials like the coloring pigments and sometimes some cell wall contents, suberenes, waxes, some higher proteins which are easily dissolvable in the protein, in the lipid loving solvents. So, lipophilicity of the sample, if the sample is lipophilic, then we need to choose the solvent so that the solute can easily dissolve into it. If the solvent is lipophilic and the solute which is desirable for extraction is little lesser in lipophilicity, then we change the ratios, and if we want to extract some material which is actually water loving and it does not the cell walls are lipophilic and the polar solvents are not able to penetrate, in such cases first we remove these interfering substances like color pigment, cell wall, using a petroleum ether like substance solvent, and then we can easily extract the desired component. So, moving to the next topic. So, the next topic is maceration. So, maceration is a method which is a closed system method, and here I would specially mention that it is cold system as well, one of the most ancient methods which is also favorable for the drugs which are unorganized in nature. So, this process, or in this process, crude drugs from either crushed or pressed plant material or animal parts are soaked in suitable solvent for several days with occasional shaking or stirring at the room temperature. This is highly important. The maceration is done at the room temperature. According to the 1966, maceration is described as place the solid material with the whole of the menstruum in a closed vessel and allow standing it for 7 days, shaking occasionally, straining, pressing the mark, and then mixing the liquids obtained, clarifying it by subsidence or by filtration. So, here there are two terms that have to be understood. One is menstruum, another is the mark. So, menstruum is the solvent which is extracting the solute, while mark is the solid or undesirable material which is staying, it is the residue. Okay. Let us see the diagram of the macerating bottle with a lid. Here the solvent system is added to the drug material. This drug could be also macerating bottle or macerating technique is also very preferred in case of fresh sample materials which is either having high moisture content or has certain volatile components which is the best in imbibed in the cold stage, and here the ratio with which the solvent volume is selected is 1:10. That means for ratio of solute versus solvent is 1:10, and the examples of very well known macerating extracts are the tincture of tolu balsams or Peru balsam. Then so these are some of the, and the tincture of myrrh and other resinous oleoresins. These are one of the most preferred method for extraction by the organized drug. Next comes percolation. Now, this is an continuous flow method, unlike the maceration. Here first the drug is powdered, then treated with sufficient amount of menstruum and uniformly wet. Once the material is damped, it is allowed to stand for about 15 minutes and then transferred to the V-shaped vessel termed as percolator. Now, again this is a closed system where a V-shaped percolator is available. Next, once the damp plant material is added into the percolator, then sufficient menstruum is added to saturate it. It is saturated, and the indication then slow passage of the solvent is done, and the lid is placed above. Once the solvent start dripping from the percolator, the lower opening is closed. So, this maceration is done for at least 20, then followed by maceration for at least 24 hours and continued gradually till we get the menstruum of almost 1000 ml of the solution. The process is highly dependent on the flow of the solvent through the powdered drug, and it yields an product which is having higher concentration than that of the maceration process. Percolation method can also be categorized, if we want to categorize it, as cold percolation, reverse percolation and hot percolation. So, a very simplified cold percolation example could be Soxhlet, which we shall be discussing in our coming slides. And then there is reverse percolation, where the menstruum which is collected once is not the final product, but the mark which is remaining here, it is pressed almost first. Initially 3/4 of the sample is collected, then mark is pressed, another 1/4 is collected, then this is again reduced and then finally added to the first percolate, and the final product is then obtained. So, and this cold maceration, it is very much similar to the maceration. Moving to the next, there is also a process termed as modified percolation, where the solvent is modified by dilute alcohol specially, and here the strength of the alcohol needs to be in such a way that it does not affect by the concentration of the extract. Now, here first again it is similar to that of reverse one, where percolation is continued and first quantity of the percolate is collected and set aside, and then subsequent quantities of the percolations are collected, and lastly the first volume of the percolate is added to the next. Okay, moving to our next method. So, this is Soxhlet extraction, and here you can see an assembly which consists of round bottom flask, extractor and a condenser. So, here the system is any capacity of the round bottom flask which is compatible with the complete assembly, same diameter of the these are compatible. Almost 2/3 of the solvent is taken. This is heated and the solvent vaporizes and goes through the this particular tube, vapor tube, and reaches to the condenser. Here the cooling happens and the hot solution then reaches to the drug material. Now, this drug material is kept in a porous chamber or thimble, and this hot condensed liquid comes and macerates the plant material in turn. Here there is kept a glass wool which does not allow the plant materials to obstruct this siphoning tube, and then once the complete maceration or extraction is done, the level of the extracted solvent raises, and once this raises to a siphon level, it completely drains out and reaches back to the RBF. Now, this return fluid is having the extracted phytoconstituent as well as the solvent system, and again it gets reheated and then reaches to the condenser, and this cycle continues. So, one complete cycle is also termed as one siphon, and such 100 cycles are done to have the to extract the phytoconstituent. Then what is done here, it should be in done for the heat thermostable substances. Moving to this, so this apparatus is designed, and it can be shown here that how the mechanism works on. We do add certain porcelain chips to avoid the bumping of the fluid, and Next is the microwave assisted extraction, which works under similar principle. The microwave increases the pressure and temperature of the vessel kept in the microwave, and the cell materials dehydrate and release its content into the system. Similar is the sonicator extraction method, where sound waves of 20 gigahertz sonicate this, and the cell material releases its content into the solvent, and this is generally used for small scale separations. Then we have this expression of oil. This is generally a mechanical type, where volatile oil is removed by a sponge method, and then usage of Soxhlet method for citrus peels, and mechanical or hydraulic pressures for or greater centrifugal or pressure is applied to get the citrus oil extract. You can see this pin. This pin will puncture the glands of the citrus fruits, and the oil would be released into the material, into the solvent, into the collector. So this is termed as Soxhlet method. With this, we shall wind up the extraction of the plant materials. Thank you.